Let's start. Don't forget to put your presents, absent. Make sure that it's recorded. And yep, welcome to this course. So we will continue to learn about data structure. So there is no book is really complete to put every data structure in bioinformatics and genomics. So we select what is important for bioinformatics and functional genomics because bioinformatics is much more wider topic like protein folding. Genomic is much more focused on the dogma from DNA, RNA, and protein, but bioinformatics is more biology and more wider. So in chapter 4, the chapter is ready for you to comment or to add feedback, and I need help from all of you in this semester to test the code, remove something that is not necessary in the textbook, yeah, because we want to use this textbook for another same lecture next semester. And so let's discuss about this chapter, and I will need help from Fadin later on to introduce you about Seqera platform. So Nextflow come up with the platform that you can just reuse the pipeline there and have user interface. So you can click, drag, and click, and your pipeline is running. I will need help later from Vadin after the chapter 4 to introduce Seqera platform so you can get started. So Seqera platform is like this, have user interface, not code base. It's a low code platform, so you can up and run, reuse pipeline, have a report, everything that is standardized by Seqera platform. Seqera is the company who make Nextflow. Is it funded by Meta or Mark Zuckerberg? They have a good mission to produce the world's largest pipeline for bioinformatics. So it's good for you to be exposed into this knowledge for industry level tools and platform. So let's back to chapter 4. So chapter 4, please read and please comment if you have comment and help me with the code. Okay. So Trianto, Majere, Jonathan, need you to run the code. I hope you have no problem with the code anymore. Most of the code now is written by the AI. So as long as you understand what you want in terms of mathematical abstraction, algorithm concept, you can always prompt, yeah, to get the good quality code. There is many AI. I think GPT and Cloud is leading about code generation. So if we compare Cloud and GPT, I will say Cloud is better in terms of code generation compared to GPT 4.0. But if you use GPT o1 and o1 Pro, is comparable between Cloud and GPT. Okay, but GPT o1 Pro is paid. So if you don't have paid GPT, you can use Cloud. But free Cloud is limited token. So maybe you can only prompt very few code per day. And how to prompt? Let me start from there. So imagine you understand the mathematics. You understand the algorithm that you want. So you can use many framework for AI prompting for code. Let's start with CARE. Explain the context, like I'm bioinformatician working in blah, 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 blah. And then I want to make a code for my research. Okay. And then give me the code. Okay. Sometimes care is not really good for code because you have to give example. But I like to use CoStar. I said context. I want to make a code to do blah, blah, blah. The objective, we want to have the code that is working well in production, can run with Nextflow. The process is separated task in Nextflow or process in Nextflow. Give the Nextflow file. You can mention the style of the code like object-oriented. You can mention also the tone of the code, like the code should be clean with documentation, blah, blah, blah. You can mention audience and you can define the response. You can also use RACE like role, action, context, expectation, or task. Okay. TAG, task, action, goal, is very simple to get the code. Problem, action, result, also very simple. Or so build your prompting skill from this framework to help you to generate the code. Okay. If you can generate the code, then you can spend more time with mathematics and algorithm. So in section 4.1 is introduce you many genomic data structure, especially functional genomic. So because of the uncertainty of the data, usually they use many data structure. One of the commonly used is position weight matrix. This data structure is to represent the motif of the DNA inside the RNA or DNA. Okay, so it's capture nucleotide at its position. Okay, it starts from base probability matrix, and then in every position in the motif, it has probability for nucleotides like A, C, G, T, blah, blah, blah. Okay, how we capture this in terms of probabilistic data structure? People are using position weight matrix. So it's weighting or scoring matrix from the weights. So we can say that this data structure is like assessing likelihood of a motif appearing at the given location in the DNA. Okay? Because DNA is very long. So the motif is a specific location is appearing or not. So we can store that information or that score or that weights inside the equation 1. For example, if the score is like this, yeah? So I hope you understand the math here. So if you don't understand, it's very easy now because you can copy here, pass to GPT and ask him to explain more detail step by step, okay? So another one is hidden Markov model. If you learned about this before, it's good, especially the keyword Markov here. So Markov is a mathematician that he said that the future probability is only depend on a few instance or occurrence in the past. Some people say Markov property is like the future is independent of past given the present. So it's not depend on too many past data or historical data, only few like one Markov below or two Markov in the past. Okay, so if we say hidden Markov models, is the idea of the sequence of observation, but it's not really depend to long sequence in the past. So hidden Markov model assume that the existence of latent state, hidden state or latent, sometimes we call latent, sometimes we call hidden state. And Markov property dictates that the future state is not really depend to whole states in the past. Yeah, in a mathematical term, called like this. So we can also use Markov hidden model or hidden models, or some people use Markov random field. So Markov random field generalize the Markov property to higher dimension, and MRF is also commonly used, but in functional genomics. So the difference is probability distribution in MRF is factorized using this equation. And from statistical properties perspective, let's say in many single cell organism, uh, it used some probability distribution like Poisson or negative binomial models to represent the counting of RNA sequence. Okay, so assess the mathematics until you get some common sense. Don't really too much on how you implement hidden Markov model, how you implement Markov random field for bioinformatics, because AI is better than us to do that job. Okay, so in this book also you get some information how this motif modeling is happen in this data structure. Okay, some use case for regulatory elements like enhancer, silencer, promoters, etc., including the transcription factor binding, how we use the data structure for that. How to implement, for example, from FASTQ raw data from sequencing to position weight matrix. So this task is very common from machine like Oxford Nanopore or Illumina, and then you get the raw data called FASTQ, and then build your position weight matrix and your Markov random field data structure. Remember that data structure is the only way we can put complex data into computer memory. So you have to choose data structure from big file like FASTQ, which data structures you want to implement. So please assess this code is running well or not, but this is a very simple next flow with one process, build MRF, build position weight matrix and Markov random field. The input is actually FASTQ file. Okay, make sure that this script can locate this FASTQ file. If not, you have to prompt again to AI that the location of the FASTQ file is in this position. So this is the next flow file, and then the output is text file. Okay. In this code, in this process, is also compile the Rust code. So you have to assess also that in this code where the compiler Rust cargo build store the output file, and then you structure the code like cargo new, and then create that, and then you add one main file main.nf, and then this is the code for building your position weight matrix and also your Markov random field. So the algorithm is well known. So first, for position weight matrix, you have to expect the same length of the sequence. Okay. Taking care this length is very important for this algorithm. And then the algorithm will initialize the count, and count the nucleotides per position, and convert the counts to probability. To convert the counts to probability, of course, the algorithm use position-wise normalization that explained before. And for random Markov random fields is the same. It should has the logic to make a count, but it used some probability distribution, transition probability distribution. And if you see hash map here, that it convert the counts to probabilities, but before it used hash map. Is another data structure in computer science to maintain a key value pair, And then this is the main function. I hope you can read the code quite obvious. So you can run this code without error. And if you find some error, you just ask again to the AI and then fix the code for you. Okay, let's move to gene regulatory network. So gene regulatory network is very powerful structure. It's a graph-based data structure. So I hope you already learned about graph, yeah? So graph is data structure contained of node and vertex. And graph can be very complex structure, can be directed acyclic graph or cyclic graph or many graph. So, but graph is very well-known data structure for many tasks in computing. So let's take illustration of this GRN. So for example, this in this picture, as you can see here, is a multidimensional interplay transcriptional and post-transcriptional regulation. This is the whole biological process actually, right? So in the central region, the transcription factor TF is a blue one, bind the regulatory elements, okay? And then resulting the mRNA here, you can see in the picture. And there is also microRNA from target 1, from target 2, creating the mRNA. How you can describe this process, biological process, into a data structure that can keep the information about processing and the vertex, yeah, the relation. Graph data structure is the best way to do that. But how we can use graph to implement this kind of concept in genomic case, yeah. So here we explain that many concepts like implementing graph using adjacency matrix, okay. Adjacency matrix is a graph construction algorithm to build the graph from the raw data, okay. And also there is some weighting or scoring mechanism, like let's say this weighted gene co-expression network analysis, you have to understand. And there is also information-based algorithm to construct the graph, like equation 8 here. This is what's so-called the algorithm for reconstruction accurate cellular network. Okay, so this is like processing from data to make a graph. Yeah, and to demonstrate this again, there is a next flow code to build. Yeah, there is a process called adjacency matrix to build the graph, and you have to make sure that the code is running. Pipeline.nf and the adjacency matrix code implemented in Rust. In some case, you can implement also with other languages like Python, but in this book, let's focus on Rust because Rust can bring you performance, parallelization, concurrency, which is hard to implement in Python. But if you prefer Python, there is always Python binding for concurrency and parallelization. Like, for example, Ryan. Ryan is a concurrency library. Of course, you can have many similar library in Python. NDRI maybe in Python is NumPy. Random in Python, but we call ran in Rust. So it's all very common library. So you can implement in Python as well. But we choose Rust because we want performance. We want to have a very fast operating pipeline. Okay. So in this section, we hope you as a students can implement your own graph based on the requirement from the biology. Of course, there is many other techniques hard to put in one book, complete book, but if you have the context, back again to your prompting technique to asking the code that you want. Yeah. This book is just a baseline, so you can have enough common sense to build your code. Okay, let's next move, motif discovery and regulatory element identification. So this is how we turn on and off the regulatory element inside the DNA or the RNA. And of course, there is many algorithm inside here. So motif discovery is like searching on the string. Like, can I search a string, long string, or long genomic sequence? And the most common algorithm in computer science for this called KMP or Knut Morris Pratt algorithm. Another algorithm is Rabin Karp. Yeah. So given a string, can I search some motif inside the string? That is the problem statement. Consider this array S is genome S, and each entry in S represents starting index of the G. So once you put into data structure, let's say you use position weight matrix data structure as a probabilistic framework to describe your motif, and you can view it as a matrix, okay? And then you can score the nucleotide at one position and implement that algorithm. Motif can also discover in other in other data structure. Yeah, but let's say for hidden Markov model, let's say we store the data in hidden Markov model. Can I search inside? So this is sample code for motif search inside the hidden Markov model. Okay, I don't give the next flow because it's just like searching algorithm. You can also implement your Knut Morris Platt or Robin Karp algorithm by changing the prompt. Yeah. So this is using position weight matrix and searching the motif inside. If you want to implement KMP, you can also do. Yeah. So it's all about searching the motif inside the text, inside the algorithm here, inside the data structure. Okay. And lastly, I give also another sample with FASTA file for motif search inside the FASTA file. Okay, the next section cover about data integration and algorithm. So sometimes we integrate many algorithm, yeah, many data. So as the pipeline grows, we need more algorithm to do our task. For example, in epigenomic data analysis, sometimes involves some processing signal from other data. So we need to equip us with many other technique for data integration. So in this section, it explain how to integrate, assuming that you already have data and you put in data structure like hidden Markov model. And there is another code sample just to ensure scalability of the code. Always think of Rayon, include Rayon in your code to make sure that the code can be parallelized well. Okay. I don't have to cover line by line of the code, but you can read and try to learn together with AI. AI do better in translating your idea or common idea into code, translating concept to code. You will have to do your research, reading more paper, looking on creativity. But now AI is better doing that. Let's say you have 10 papers on one topic you want to research, upload to AI and find out the difference, summarizing the difference between papers. AI do better than you. But there is area that we are better than AI, especially if this is not related to knowledge that in the book or paper. Okay. If this is related to other common sense that is not in the book or in the code or in the paper, so you can add your common sense over there. What about transcriptomics, the process from the RNA transcripted to the protein? Okay, there's also many algorithm here. So if you see the workflow of transcriptomic analysis from the plasma, RNA extraction, purified RNA, and then we store in data structure and have to perform some analysis. So there is something happen inside the machine, and after that you will have the RNA data, and you have to store the data. Okay, so RNA process is explained here based on this picture, and then you will also deal with many graph algorithm because this process contains some node and vertex or process and relation that is best to put in graph instead of you put in the sequential data structure. So most of people using graph for uh, storing this kind of information and computing this information. Once you know that they're using graph, you can always asking for use case scenario. Let's say you want to see how to build and merging partial splicing graph that represent gene transcription. You can ask the code and ask the code more specifically to be, let's say, parallel code or high performance code using high performance computing, or you can say that audit the code for better performance. And this is the prompt come up with Rayon. Rayon is widely used. You can see how to slice the graph here. Okay. And also single cell functional genomic. Sometimes there is some specific data structure like compressed sparse row CSR or compressed sparse column. Here the explanation, and also some mathematical concept here like PCA. PCA is dimensionality reduction technique. So if you have a large, let's say, matrix, you want to reduce the size, you can do PCA or you can do SVD, matrix decomposition, to reduce the size of your data. Okay, and give you also the example how to construct a graph based on nearest neighbor of the cell and implement KNN graph. And inside the graph, once you construct the graph, you can search using breadth first search. This is graph algorithm to navigate or to travel inside the graph. Okay, so another code sample using compressed sparse row. So mostly in this chapter, explaining common data structure for basic functional genomic tasks like searching, motif, and mapping the expression or functional variant discovery. Yeah. So this is the last section, I believe, explaining the EQTL. So expression quantitative trait locus is a mapping function, yeah, in functional genomic to link the genetic variation with changes in the gene expression. Okay, so many papers talk about this process, this algorithm. Yep. So EQTL can be understood simply like which genetic variant or SNP polymorphism affect the expression level of which genes. In simpler terms, picture each SNP as a small difference in DNA and each gene as a measurable quantity of mRNA inside a cell. So EQTL signal that individual carrying certain variant of SNP are likely to show higher or lower mRNA. Please read carefully the scenario, build your common sense, because that will more better for you instead of memorizing the algorithm. As long as you know that the algorithm run use, for example, graph algorithm, you can always ask which algorithm is better than which one. compare the complexity, give me the best one, and implement the best one using the language you like. But understanding the process, the biologic process, is very important to get your common sense. And then give you the code here also to implement ETL pipeline. This is still using Ryan, but there is another crate here called StatRS. This is a statistical crate for you to implement many probability distribution, like student T, continuous CDF. So StatRS is a commonly used statistical crate for generating probability distribution. Maybe inside the code, there is some requirement to generate some probability distribution. Okay, so in the summary, as summarized here, in reality, if you work in bioinformatics industry, you have to be able to parallelize, implement concurrency, implement high performance code, because we are dealing with very big data. Yeah. Other option is to use library, to use pipeline that already proven in the community. Because if you implement by yourself, then you may have mistakes. If you use other people code, they maybe already have that mistake and they correct or they fix the mistake and get proper pipeline. So Nextflow come up with NF Core is a collection of proven pipeline from the community. So there's many pipeline here. Okay, you can search pipeline that already released or underdeveloped or archived or no longer released, being used. But there's many pipeline here. As you can see, total about 126 pipeline in functional genomic, like genomic QC. Okay, many. Yeah. Okay. So people reuse instead of build from scratch. And once you get, let's say, one pipeline you want to reproduce, you can simply ask ChatGPT or Cloud, how can I run this pipeline? Yeah. Let's pick one. Let's say variant benchmarking. So this pipeline is to evaluate and validate accuracy of variant calling method in genomic research. Yeah, so many variant calling techniques because it's a probabilistic techniques. So how we benchmark them? So this pipeline is to evaluate and validate or benchmark many pipeline techniques. You can see here how it works. It's a very complex process. Imagine if you don't use Nextflow, how you can guarantee that its process running well, and how you can make, if the process fail, it have to resume and back again. Okay, so this workflow is best to implement in the good tools. Before Nextflow, we have many other tools like SnakeMake or other tools, but this is, I think, the best option currently, because people can work together as a community, can share in one place. Okay. So each workflow have the usage, how to call it, yeah, how to prepare the sample data. Okay. And each workflow pipeline have parameter. What is the input to enter the complex process workflow inside the pipeline? Understanding this from the documentation is very important. Okay. And what is the output and how to assess the result? Okay. So your option is you have infrastructure in the cloud and you run this workflow manually, or you can use Seqera platform. So Seqera have AI features as well now. So if you working with Nextflow, you can install the next flow in your cursor. Yeah, I think they already have update here. Okay, so many new features as you update here. You can have AI features. You can ask directly to Seqera AI to generate the next flow code for you. Yeah. So Seqera also hosts the pipeline like from the NF4. So you can run the pipeline directly in the platform without coding, as long as you can pay the cost of training. So you can use this. But I believe Fadin is there.
Yes, sir.
Okay, can you give the close this lecture by give introduction to this platform?
Yes, I can.
Do you have a complete subscription for Seqera, Fadin?
No, so for Seqera, for the personal use, we have to connect into our own cloud, for example, Azure, AWS, or GCP. But in Community or Showcase Pro, the users can try to run the available pipeline in here and
Uh,
launch it in here. So it will be,
can you show to them?
yes, I can.
Okay, so thank you very much, Fadin. So it's very helpful for you to utilize Seqera if the pipeline you want to reduce. But as a master degree, maybe you have to modify the pipeline, change some process. There is where your Rust knowledge will be very important to improve the performance. Okay, please, Fadin, continue. I will stop my lecture here and please learn together with Fadin regarding Seqera platform.
Okay, thank you, Mr. Isman. I will share my screen first. So in Seqera is actually a platform to run, to build, deploy, and scale our experience in Nextflow. And in here, you can sign in with your GitHub account. And if you click dashboard, it will redirect you to the cloud of Seqera. So in here, if you want to try the Seqera AI, you can click on here and we can chat with that like you can do with ChatGPT. For example, we can ask what pipeline can I use for variant calling. It will answer you based on the Seqera search. And via called Seqera.io. So it have two different things in here. If you have a personal account that have a compute environments, you can add compute environments in here and choose the platform to run your experiments. For example, if you have Azure or if you have Kubernetes or have Google, you can link in here to compute environment. So for demo purposes, the Seqera have the community in here. It has showcase. And in here, the community showcase, we can use the pre-built community pipelines using 100 free CPU hours. And in here, you can also see the guide how to run the RNA analysis via platform cloud in here. And as you can see in here, in Launchpad, there are several pipelines available that already pre-configured in here. So it's actually linked based on the GitHub of NF. For example, this one is for the GitHub for NF Hello Genomics, and this one is for NF for protein structure. So there are few examples that we can try to run and see how it will work in here. So if we click on the launch, it will pop up the configuration of how we want to run the NF Hello Genomics. So in here, this already pre-configured, or also use the sample of the BUMS file, and it's stored in the AWS cloud for Seqera. And we can the general config is already pre-set up, so you don't have to change anything. And in here in run parameters, if you have a parameters for specific file, you can add in here, but we can use also the sample file. And that's it, we can launch. And the runs will be submitted into the cloud to execute it. And in here, we can monitor and inspect the details of workflow execution in your workspace. So because it is the community showcase, others people pipeline that run inside the community showcase also will pop up in here, so we can track on which pipeline that you run previously. In here, I already run the NFL Genomics, and it is succeeded, so it have the status of each pipeline. And I already also run the previous one for NF type, and it's still running. And the latest one that I showed you earlier, it's being submitted, so we can check on in here. And I will show you the result in this one. So it is pre-configured by Nextflow. That's why the name is Sleepy Hilbert. And here we can see the status, which is there any task that is failed, or is there any task that is being running, or is there any task that is being pending. In here, we can also track the process. So in this, if we can see in the GitHub, the NFL Genomics is demonstration purposes, the basic genomic analysis using GATK, Genome Analysis Toolkit. In here, we use the PCF tool stats, and it will show the result. And we have to use the BUM and also use reference FASTA file in here. So in these things, we have 5 processes. First one is some tools for indexing, and the second one is the ATK haplotype color, and the third one is also the ATK joint genotyping. The fourth is BCF tools, and the last one is multi-QC to check the qualities of the sequencing. In here, we can also check each process and the attempt, and we can see the result of our pipeline in reports. In reports, we have the multi QC reports that we can see the reports in these para preview reports, and we also can download or open in new tab to give a better viewing. So we wait. Okay, in this is the result from previous pipeline that I ran in MultiQC. So in MultiQC, it is a modular tool to aggregate results from our bioinformatic analysis that if you want to run it, we can compile it into a single report. So in here, we can see the BCF tool stats, and we can see the variant quality count, and in here it's already available everything that already running from our pipeline. And that's it for the multi QC. And in here in the runs, we can see our pipeline that is being running, and we can see also the succeeded pipeline that already being run from others people. And in here we can also see the data sets. the datasets that available to run inside the Seqera platform. And this is the data explorers. This one is from 10,000 genomes. This is placed in the S3 buckets via AWS. And this is the compute environment available to run the workflows inside in community showcase. So if you have any computing power available and want to run the bioinformatics process, you can tie up your compute environment into your personal cloud in Seqera. Otherwise, for demo purposes and for exploring, you can run it in the community or showcase cloud for in Seqera. And because this is we have the 1000 free CPUs hours to pre-build to explore pre-built community pipelines. I suggest that you can run and follow the guide to run the first analysis in here. So what is the difference with the Nextflow? So basically the Sequoia is um where we want to run the execution of our pipeline. So there are few options to run it. Like we can do that in our terminal if VS Code, or we can run it in here by connecting in Seqera platform for more intuitive and user friendly because they have the UI for how we can configure each pipeline. So it's a matter of preference how we can run it, but I suggest you to explore because there are so many available tools in here in Seqera that we can use. Okay, that's it for today's class in bioinformatics in the week 4. Don't forget to put your weekly task for each chapter in your GitHub and also put the GitHub in the app presence form and also to fill in the presence form in each week. And don't forget to submit it on time and it will also be calculated into your final grades in the end of the semester. Okay, is there any question that you want to ask?
Hello.
Hello, yes, ma'am.
Sorry, ma'am. Maybe I'll be taking you back a bit. So I'm using a Cursor for my coding. Do you mean that for next flow pipelines, I can use Seqera instead of Cursor?
Okay, so for the Nextflow pipeline, because this is for Seqera, it is for how we can build and deploy it, and we can use it in the inside in Seqera platform. So this is the platform which can have to end and also collaboration, and also we can track on of each our cases, so we can use Seqera. In here, yes, we can use Nextflow, all Nextflow pipeline, and also our pipeline. If you have in here, you can create a pipeline, name the pipeline, and you have to have the compute environment first to be able to run if you have your own experiments in here, but otherwise you can explore in here in the community showcase. So the Seqera is actually after you have the pipeline available already, you have finished your pipeline in Nextflow and you want to run it not in your laptop but in the Seqera cloud platform, you can do that in here.
Okay, I got you. Thank you, ma'am.
Thank you.
So for my publication, I did my work and I'm now realizing that two branches, main branch and master branch. So I put my experiments in master branch, which is not a default branch in the
Okay. Let me
story.
first for your GitHub, right?
Yeah,
Okay.
I'm still facing challenges in that, but I think very soon
Okay.
I'll be okay.
Let me share my screen again. Hmm. Okay, I already founded the branches. It is the master in here, right?
Yes.
Okay, so if you have two branches in here, you have to sync up, but to get the changes in the other branches to the main branches, but it's okay if I can look up in here and you can um put your works in here if you have difficulties to merge into your main branches.
Okay. So is it possible that I can rearrange like what Mr. Triando did and put it in the main branch?
Yes, 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 it is possible.
Oh,
So, um, how to do that? In here, I think you have to just how to merge GitHub branches. In here, how to merge two branches in GitHub.
oh, okay, okay, okay.
So it is because GitHub is for version controlling. This one is sync how to sync your branch in GitHub. This one is for if you have GitHub desktop, you can pulling and also merge another branch into your project in here. So, but if you have troubles, for example, like you cannot maybe you want to put it the arrangement like sorry like in here.
Yes, like that one.
Yeah, it's okay if you want to create a new repositories if it's easier for you to rearrange everything, and you can move and copy your existing works in your new repositories with um better structure for each chapter, have each experiments, and there are also explanation and also have as the file needed to run in each experiment. So it's okay if you want to maybe create a new repositories and update your GitHub repositories in the presence form. It's okay also. No matter choose what is the easiest from your side, and I can check your works later on.
Okay, ma'am. Thank you very much.
Okay, thank you. Okay, so if there are no questions, I will stop this lecture and see you guys in next week for continue to the next chapter. Today we already learned about Seqera platform and also for chapter 4 about data structures and algorithms for genomics. In the next class, we will continue to learn for chapter 5, which will be sequence analysis and processing with RAS. So don't forget to submit the task and also update the GitHub. And see you guys again in next week. Thank you.
Thank you.
Thank you.